All
Yep,
right,
you're recording and it's 3 o'clock.
perfect. Hi, everybody. Thanks for joining us to this webinar series. So that's the Research and Winemaking webinar series that we started a few years ago. But thanks for being with us today. We know it's a really busy time in the year, so thanks for being with us. We hope you will enjoy the webinar today and you will develop knowledge and skills that you can use in the vineyard and the winery. So I'm Dr. Aude Watrelot. I'm an assistant professor and extension specialist of enology at Iowa State University. And I'm co-hosting this webinar with my friend and colleague, Drew Horton, who is a research specialist of enology at the University of Minnesota, Grapes Breeding and Enology Project. So before starting the webinar, there are a few things that we would like to mention. That's about the recording. So this webinar is being recorded and the previous recordings are available on YouTube following this link that I will be sharing on the chat box. Then if you have any questions during all the presentation, feel free to use the chat box or the Q&A box or unmute yourself if you have any urgent questions. And then at the end of the webinar, you're going to receive an email to complete a survey. So please take 2 to 5 minutes to complete that survey. That's really important for us to know what to improve and what to change, just to make sure you have everything you need. So today we will have 2 presentations. The first one is going to start by Dr. Richard DeScenzo from ETS Laboratories in California. He will present and address the protein stability or instability and haze formation as it impacts the wine production process. And this presentation will be followed by a presentation from Dr. Belinda Kemp from NIAB in UK. So she's far from us, I think. It's pretty late for her. But she will present some of her research on proteins involved in sparkling wines. After the presentation, we will discuss and answer any question from the audience with grape and wine industry members, who is going to be Zwink from Creek Winery in Iowa. And we were supposed to have Ben Banks from Sovereign Estates in Minnesota, but he was unavailable, unfortunately, today. So, Drew, let's start with the presentation of Richard.
I will. I'll start by introducing Dr. Richard DeScenzo, who is the microbiology group leader and co-general manager for ETS Laboratories in California. ETS provides a range of chemistry and microbiology analyses to help clients in the winemaking process. In addition to providing microbiology support for the wine industry, Rich focuses on helping clients with a wide range of other winemaking problems. A little about Rich's background: he received his master's in plant pathology and a PhD in plant biology at the University of New Hampshire, and conducted his postdoctoral studies on disease resistance genes in barley with the U.S. Department of Agriculture, ARS, at Iowa State University. He then spent 10 years conducting research on grape genomics, development of molecular diagnostics, and fermentation monitoring in the wine industry prior to joining the ETS team. Richard, the floor is yours.
All right, thank you. Let's see if we can make this work. There we go. Looks good so far. Ta-da! All right. So we've conquered the first and most difficult part of a webinar, and that's getting the presentation up on the page. So I'd like to thank Aude and Drew for giving me the opportunity to speak with you today. And we're going to talk about heat stability, which is, as long as people have been bottling white wines, there have been issues with heat stability. It's not something new, but I'd like to tell you about some of the new things we're doing with it and just overall discuss the problem itself. So without anything else, we'll just get started. So heat stability usually refers to the formation of haze in proteins that are exposed to high temperatures, in wines exposed to high temperatures. And you all know, especially if you make wine and you sell it and someone leaves it in the car on a hot summer day, quite often, if the wine isn't heat stable, they're going to come back with a cloudy bottle with a precipitate. So having that haze form exposed to, when the wine is exposed to high temperatures, is a very real problem in the industry. Now, haze formation occurs when colloidal particles, and that just means when little pieces start sticking together and they get bigger and bigger. And so these usually contain protein, that's a primary component, but they also contain phenolic compounds, polysaccharides, and other factors, and they all just start sticking together and they form this colloidal particle. I'll show you some of that under the scope in a minute. So the important thing to understand is that although protein is a primary component, it is not the only thing um, uh, that takes, that plays a role in the haze formation. So it's related to the amount, but it's not the only thing. So you can have a given amount of protein, and if you're lacking other factors, you won't have a haze. But it certainly is the primary component, and the one that we focus on when we try to heat stabilize wines. So I would say, other than oxidation, I think is probably one of the biggest problems in wine and wine stability. But protein haze is one of the key stability issues. And if you're going to ship your wine anywhere, to wine club members or to distribution outlets, the wine has to be heat stable. So what causes heat instability? Okay, what causes that haze to form in the wine? Well, wine has proteins in wines, and they can range from very low levels, 30 milligrams per liter, up to 3, 4, 5, 600, depending on the varietal. And proteins have a structure, and we call it a tertiary structure, and that's just a single protein moiety. You can have different subunits all stuck together. So proteins are very complex. And the way they are formed, either coiled or subunits all stuck together, determines their ability to function as in this case, mostly enzymes. Okay, and so when you heat it, it becomes denatured. It opens up. So instead of having this nice globular or tertiary type structure, it opens up into these linear strands, chains of amino acids. And when that happens, other things can occur. So heat causes the protein to denature, and then other things start sticking in. And one of the primary, what they used to call X factors, but factors that they've shown to be play a major role in this, are sulfates. And sulfates are found in all wines, at least if the wine has SO2 in it, and any transition metals like copper or iron, then you will have sulfates. So sulfates bind to this opened up protein, and they can cleave disulfide bonds and do other things, but they pretty much fix the proteins, that the protein is stuck open in that denatured phase. And then all kinds of other things, other proteins, polysaccharides, phenolic compounds can start sticking in there. And this is how you start getting that growth of that particle from that initial small little strand of amino acids. Things start getting together, and you end up with particles. And this is taken with just standard phase contrast optics at 400 magnification, and that glob in the center there is about the size of, say, 5 or 6 yeast stuck together, to put it in context. And that's what it looks like if you're looking through a phase contrast microscope. If you use change over to differential interference contrast, which kind of illuminates from the side, you get almost a 3D effect, and you can see that same particle now is looking like little globs and balls, and they're just all stuck together. And so what happens is, as those proteins and sulfates and other proteins and other compounds start sticking together, they're very sticky, and they all start glomming together. And when they're maybe 1 to 3 microns in diameter, they stay in suspension pretty well. And that causes the haze, but they continue to stick together until they get, to be 10 to 15 to 20 microns. And then gravity takes hold, and they start to settle out. And there's a lot of factors determining how quickly they settle or whatever. But if you ever picked up a bottle that's a wine that's been heated and is unstable, if it's brand new, you'll see a haze, but if it sits for a while, you'll end up with a sediment down on the bottom. And so that's the protein haze component. That's what we're talking about. So what proteins play a role in heat instabilities in grapes, and where do they come from? There's been a lot of really good research done here. starting in Australia, and just a number of places, Italy, France, in the US. So a lot of work done on this. And it turns out that the proteins that play a major role in heat instability are what we call grape pathogenesis related proteins. And that's a big word for just saying proteins produced by the plant in response to being attacked or wounded. And so these proteins are very, very stable. They're produced in response to both biotics, so something like fungal or bacteria, or I guess you could even call bird or bee damage a biotic stress, and then also abiotic stress. So anything that stresses the plant, you can get these proteins formed. And the two primary ones are chitinase and then thaumatin-like protein, or TLP. And these are part of the plant's defense response, if you will, almost like the plant's immune system, although not quite the same, but it's a response to infection or damage. And so obviously, if a berry has a botrytis infection, then you're going to see upregulation of these proteins. Now, they're expressed what we call constitutively at low levels all the time in the berry, but if there's any type of an infection or damage, then they become what we call upregulated, so you have higher levels. What a lot of people don't think about is what happens when you go out in the vineyard and you drop fruit or you do canopy management. This is actually wounding the plant. And I'm trying now to work with some vineyard management companies that we can look at the levels of these proteins in the same varietal, but with vastly different canopy management routines or fruit dropping routines. Because like I said, you don't hear the plant screaming, you don't hear the vine screaming when you're cutting off and fruit or dropping clusters or doing canopy management, but you are wounding the plant. And so I have this theory that the more intensively managed the vineyard is, the more of these proteins will be present. I could be completely wrong, but that's my theory for now, and I'm sticking to it. I hope to have some data for that in the next few years. So chitinase. Chitinase is a very important, if you will, the first step in this protection process, because the cell wall components for fungi, yeast, and for that matter, lobsters and insects is chitin. So chitin is that primary cell wall component. And what chitinase does is it hydrolyzes chitin, resulting in damage to the fungal or yeast cell wall. So that's the first step. Let's damage the cell wall, and that way we can get through to the membrane. And like I said, they can be expressed at low levels all the time, but dramatically enhanced by both abiotic and biotic factors. The next one is the thaumatin-like protein. And these proteins, thaumatin-like proteins, are classified as osmotins. So they've been detected in a number of plant species. And what they do is they enhance membrane permeability. So they cause a rupture of the fungal membrane. So the first one, the chitinase, chews up the cell wall to damage it. And then the second one, the thaumatin-like protein, helps pop the membrane, causing cell death. So this is the plant's response, okay? So these proteins, by the way, are incredibly stable. Pathogenesis proteins in general have quite a few disulfide linkages, and those disulfide linkages make it very resistant to things like ethanol or other things that normally you'll end up with protein degradation. So these are really the only proteins, the pathogenesis proteins, that make it from the grape into the finished wine. There's a couple of other minor pathogenesis proteins, some invertases and glucanases, but the main two are the chitinase and the thaumatin-like protein. So how do we measure the proteins? Well, we wanted to look at this a few years back. I was more interested in whether these pathogenesis-related proteins could cause stuck fermentations, because after all, yeast are fungi. And so we developed antibodies targeted to the proteins. We developed what we call an enzyme-linked immunosorbent assay. So if any of you were doing COVID tests over the last 3 years or so, you're using an enzyme-linked assay to detect the presence of the viral coat protein or spike protein. And so it's a similar method where we're using an antibody which is specific to these proteins, the chitinase and the thaumatin-like protein. And we validated these assays, and they work very nicely. Essentially, with an ELISA assay, It's widely used. It detects the presence of this protein and of these two proteins in particular. We can get down to 0.3 milligrams per liter, which is pretty sensitive. And essentially, if you go from left to right, we have what's called a capture antibody in the plate. You add the sample, and so if the sample has chitinase, excuse me, or orthogonal-like protein, it binds to the capture antibody. You put a detection antibody on it, and then essentially we go through a substrate color change. And the way it works is you can look at the plate and you can see, well, where there's yellow, there's no chitinase or thaumatin-like protein. Where there's blue, there is. So very simple test, easy to do. You can run a lot of them at once. And so that was the test we developed. Just to show you, this is a use of chitinase, excuse me, of bentonite to remove chitinase from juice. Normally, we don't do heat stabilities on juice simply because, in order to do a heat stability test, you really have to get it clarified and run it through a filter, and that's a pain. So, but we did for this test just to show you. So the blue bars are the NTUs, okay, that's the turbidity, and I'll talk a little bit more about that in a minute. And the orange bar is the chitinase present. So with no bentonite added, we were probably about 23 chitinase milligrams per liter, and the NTUs were up at 65. When we successfully added across the bottom 1 pound, 2 pounds, 3 pounds, 4 pounds, 5 pounds of bentonite, you could see that both the level of chitinase protein and the turbidity came down. We found that when we got down to levels of protein below 1 milligram per liter, we weren't seeing, or I should say the NTUs were correspondingly less than 1 NTU. So shows very dramatically, if you remove the protein from the juice, it becomes stable. So this is a test we offer now to clients. There seems to be a big push in the industry to get your bentonite in up front in the juice. They feel it has less impact on things like aroma because a lot of the aroma compounds that might bind to bentonite in wine are still glycosylated in the juice, and so therefore they don't bind to the bentonite. So for a number of reasons, people like to do it up front, and this is one way of testing to see if it's effective. So how do we determine heat stability? How do we know if a wine is heat stable or not? And this is something that there's a lot of tools available. Most of these, ethanol precipitation, bento test, ProStab, tannin precipitation, the TCA and the ammonium sulfate, are all ways of salting out or precipitating out protein. And so these are, one way of doing it. And then you use usually a colorimetric, some other type of test to actually see, with the standard curve, whatever, how much protein is in there. But that's just one of the components, as I said before. Most people in the industry use a heating method, some variation of a heating method. So you can do high temperature, short time, low temperature, long time. Nobody has time to do a long time heat stability test. It's always last minute, it seems, at least from my end. They do alternating high and low. Rankin published a method back in '73, which I believe was 6 hours at 80 degrees C. And then Elizabeth Waters, with her major professor at the time, Pocock, developed a method where they reduced the amount of time. And so what we do at ETS is we do 2 hours at 80 degrees C, okay? And many people say that's extreme, but generally if something passes that Pocock and Waters test, then it's going to be heat stable even under extreme conditions. And then we do the 2 hours at 80 C, then 6 hours at 4 degrees C, then we bring it to room temperature and read it. And when we read it, what are we reading? You get a report, you get results in NTU or nephelometric turbidity units, which is just a big word for saying light scattering units. And that's what it does. We're measuring light scattering, okay? Most clients target to be below 1 NTU for stability. Most of the European textbooks indicate a value of 2 NTU or less is acceptable. And truthfully, if I hold up a tube with a 1 NTU standard and a 2 NTU standard, I can't see the difference with my eyes. So probably 1 to 2 NTU units is a good target, somewhere in there. There's all kinds of descriptions, and people will say between 1 and 2 NTU is a light haze. I disagree with that, but certainly as you get up to 3, 4, 5, it gets pretty hazy. And I mean, we've seen things come back from heat stabilities with, 2 or 3 hundredths, and those are pretty opaque. So how do we heat stabilize wines? Well, we use bentonite. And bentonite's a natural clay with an overall negative charge. Everyone knows that. Essentially, hydrating the bentonite properly maximizes the available number of negative charge sites available to bind protein. That's probably the most key thing. It is the most widely used process for heat stability. It's very important to understand that proteins have an isoelectric focus point where they are charge neutral. And if the pH shifts above or below that point, they either have a positive or a negative charge. Bentonite has an overall negative charge, so therefore it binds things with a positive charge. And I'll do this quick diagram to see if we can get through this. But suppose you have a protein, 3 different proteins. One has this isoelectric focus point at 2.8 over here, one at 3.5, and one at 4.0. If those are all in a wine with a pH of 3.5, then the protein with the pi at 3.5 has a net neutral charge. It doesn't have any charge to it. It's not going to interact with bentonite. The protein with a pi of 2.8 has a net negative charge, so it's also not going to act with the bentonite. But the protein with a pi of 4.0 has a net positive charge, so it'll interact with the bentonite. And so most pathogenesis proteins have PIs of 4.1 to 5.8, which is why with most wines we're able to find them out with bentonite. However, if you have a high pH wine, your bentonite finding may just be less efficient. So real quick, bentonite's an absorbent aluminum phyllosilicate clay, okay? These montmorillonites. They can either be sodium or calcium. Most of the time, they're kind of a blend, a little bit of both. But now they're actually pushing the calcium ones because they go into suspension easier. But the sodium has a greater swelling capacity. But always people like to do things easier. If I don't have to heat it, if I can just mix it up really fast. So there's been more use in people adopting using the calcium bentonite. However, if you're in an area that has higher calcium soils, by using a calcium bentonite, you increase the risk for increasing calcium, which of course can increase the risk for calcium tartrate precipitation. So not all bentonite products are the same or perform the same. I will tell you, no matter what bentonite you buy, and you can tell the vendors I said this, they can't argue, we've done the math. If you hot soak a bentonite, it's going to perform better than if you room temp soak it. But a lot of them now are, just pour them in, just mix them with some water. You don't have to use cold water, it doesn't matter. And they're absolutely right. It will work, but it won't be as effective. The hotter the water, the better swelling of the plates. There's a lot of use of bentonite in storage facilities where they don't want anything that leaks out to get into the water table, and especially in radioactive waste storage facilities. So there's a ton of literature on bentonite in those types of sciences. And this was something from a nuclear waste site article on bentonite. And basically says the swelling of bentonite remained constant from 25 to 50 C. The swelling ratio started to increase when the temperature was above 70 C. So the higher the temperature, the more movement, the more it opens up, the more negative charge spaces, the more effective it'll be. And I'll get off my high horse on bentonite there. We at ETS offer many different bentonites for people to use, and some of these you can see, the majority of them are cold soak, what they call cold temp, and that's because they're easy. I call them easy bentonites. People like it because they don't have to boil water, but you get what you get. Okay, so finally, we get a question a lot. Why don't I get the same results in the winery as I get on my lab report? Well, lab trials, we do them with 100 ml volume. We have a perfectly prepared slurry, and we have rapid and thorough mixing. So the bentonite is completely mixed into the wine in about 30 seconds. That doesn't happen at the winery. There can be differences in bentonite preparation and hydration time. Difference in the time it takes for a thorough mixing. Thoroughly mixing a tank is not a trivial thing. And then, you can also get process changes to the wine prior to bentonite addition. A lot of times people send it into us and they do something like add some tartaric or do a cold stability, and then they do heat stability and they get a slightly different result. But most of the time, you should get pretty similar results, but never will you achieve exactly what we get. So it's always, I tell people 80% efficacy on what we get. So finally, in closing, do you need to chase the numbers? Overall, the industry is concerned that current methods of analysis overestimate haze potential, and we're adding way too much bentonite. Well, the ideal way to determine if a wine is stable is to hold it at the temperatures you want for the length of time you want it to keep stable. But nobody has time to hold their wine for 6 months at 40 degrees or something to see. So we want a fast method. So we're looking at generally heat stability. As I said before, protein content is not always predictive, usually, but not always. A wine that passes the Pocock and Waters method will be heat stable under most shipping and storage conditions. But you don't have to chase the numbers. In many cases, it's not necessary to chase the numbers for heat stability. Someone who's making a small batch of rosé, they're only selling it from their tasting room, they can educate their clients. Then I tell them, no, don't add 10 pounds of bentonite that's required to heat stabilize it. Instead, educate your clients. Add just enough bentonite to get the most reactive protein and leave it at that. And so with that, that's a whirlwind on heat stability. And please, if there are any questions, we can address those a little bit later. But thank you very much for listening.
Thanks very much, Richard. It was great. Great overview of all the techniques or how to use bentonite and do heat stability test. That's good. So if you have any questions, please use the chat box to type them, and we will answer them at the end of the presentation from Dr. Kemp. So that's my pleasure now to introduce Dr. Kemp. Let me make her presenter first. All right. So Dr. Belinda Kemp is the group leader of NIAB, which is the National Institute of Agricultural Botany in the UK, providing scientific leadership in viticulture and enology research at the Wine Innovation Center. Belinda gained her PhD in 2010 at Lincoln University in New Zealand, studying Pinot Noir tannin, flavor, and sensory characteristics of wine. As well as a scientist, Belinda has gained practical winemaking in commercial wineries in New Zealand and the UK. Belinda joined the Cool Climate Enology and Viticulture Institute in Ontario, Canada, as principal scientist from 2013 until 2023, where she led research focused on base and sparkling wine and red wine tannin, and joined NIAB to lead the viticulture and enology research at the Wine Innovation Center. She is currently working on spur versus cane pruning of Pinot Noir, Chardonnay, and Bacchus, and her enology research includes red winemaking with a range of varieties, calcium stability, and the Maillard reaction in wine. Belinda, the floor is yours.
Can I interrupt just for a second? Belinda, can you click that icon again to expand your screen?
Oh gosh, which icon again?
You had it expanded for a second and then it reverted. Again, that little screen icon at the lower right. That one.
Brilliant.
Perfect.
Okay.
Thank you, Belinda.
That's all right. Thank you first of all for asking me. I've been here in the UK back home since April when I took over the role of leader of all viticulture and enology research at NIAB. NIAB stands for the National Institute of Agricultural Botany. Before I came here, they built, just to give you some information really, but in case anyone ever chooses to visit England, do let me know and you can come and visit us. But it's the first and it's new, and it's also the first wine innovation centre here at East Malling in Kent. There's 3 departments, so we do genetics and fruit breeding and pest and pathogen ecology and crop science and production systems. And that's our artist impression of the winery, because I haven't managed to get a drone to fly over the top of it yet. And that's the entrance here, down here on the right is the entrance to the Wine Innovation Centre. It's a bit warmer than being in Canada, I have to say, but it's interesting challenges over here. So some of this, I don't want to repeat what Richard was saying earlier. So some things I might skip over and you might have questions about afterwards. Richard mentioned the main proteins involved in protein haze, so TLPs and chitinases. There are some others, but nobody really, other than, well, beta-glucanase has been quite a lot of work on for a number of reasons, and invertase. But the grape ripening related proteins and the LTPs, the lipid ones, it's tough to find any work on those, and that's probably because they're not so important as the others. But we never know. Things might change with climate change as ripening changes. You've seen all of this lots of times, the difference in the protein haze. The thing that all our work, when we worked on Covey and Canada, was pretty much all my work was sparkling wine or red wine, predominantly sparkling. And it's been known for decades. Well, that's probably not entirely true, but it's certainly been known for years and years and years that grape and yeast protein give different things to the foaming, excuse me, the foaming of the wine, which of course is what's so important for traditional method sparkling wine. So one of the questions that has often been asked, and it was asked when I first arrived in Canada, is, does sparkling wine get protein haze? And it didn't used to. And one of the reasons why there are a few more cases of protein haze in sparkling, traditional method sparkling, than there used to be, is down to the difference in the chemical composition of the grapes ripening. And I'll go into this. So yes, the time of harvest, the ripeness and maturity level of those grapes will affect the proteins, the grape proteins that are responsible for haze. We know that the grape proteins are actually very important for sparkling wine foam and to keep that foam bubbling up for the effervescence. But I can't tell you from my research, and I couldn't find anything when I was putting this together, naming which specific out of those proteins in the last slide, which specific proteins in that list is responsible for the foaming. We haven't got to that much detail in research. So if somebody wants something to do to be able to be more accurate and be more targeted with things like bentonite use and different products, that would be a really good way to go, is identify specifically which proteins are responsible for the haze but are good for the excuse me, good for the foam. So we use a lot of bentonite in sparkling. I should say we shouldn't use a lot, but it does seem to get used in many places. Hopefully not where you guys are, and I'll explain why. This is not done on sparkling wine, this graph, but I did find it very interesting for the number of products that they used. And obviously no bentonite. The bentonite was the positive control that removed the grape proteins in this study. This is going back to 2012. And of course, the unfined ones had loads still. But if you look along all the treatments that they used, albumin, chitosan, PVPP, chitin, casein, etc., isinglass, I'm finding it really interesting that we haven't got a bit better at mixing and matching a little bit more. So there have been studies on blending 50% chitosan and 50% bentonite. But as Richard alluded to, there's loads of different bentonites, and there's actually quite a lot of different chitosans now, different molecular weights, made from different things. Like mushrooms, now it's made from mushrooms as well as sea crustaceans, and as well as Aspergillus niger. So we need as well, this is another area of research where we need to be a bit better at looking at all the different options, the mixtures, bentonite mixtures and chitosan mixtures all hooked up together. But you can reduce the bentonite use by using some chitosan. And of course, everybody knows about bentonite and aroma, as you know, Richard said, too much bentonite, and he mentioned as well calcium and sodium bentonite, which is something else I'll come on to in a second. One of the difficulties with making traditional sparkling wine I've encountered in many places is when a winemaker or winery wants to increase their SKUs, increase the amount of products, and then start a sparkling wine program. Unless you've had some proper sparkling wine focused training, what I have found is that there's been quite a lot of cases where people are treating the base wine like a still wine. So they'll add bentonite to the juice or to the fermentation. Then they'll bentonite fine the base wine, and then of course you've also got bentonite in the as an adjuvant. That's an awful lot of bentonite, and so straight away you've stripped loads of stuff out, not just the proteins that you want for the foaming, but of course the flavors and all the good stuff that we need for the aging and for the foam, for the compounds that come later, like the Maillard reaction compounds with the caramel and that side of things. So there's a lot of and this, I'm talking generally. I don't know what your sparkling wines are like in your area, so please don't be offended while I'm saying things like this. But overfining of base wines is really detrimental. Again, Richard's alluded to some of that in still wines too. But as Richard's slide showed, the sodium bentonite ratio is really important, and we're really hard water here in the UK, which means that we've got loads of calcium in our water. And so if we're adding bentonite, we're adding calcium in our water that we're using to prepare it. And then we also could be adding a bit more through the bentonite. You know, the bentonite leaves it as well. So what happens as well is that we really in the UK are focused on sodium calcium mix, so that we're not stripping the sodium so strong that it can take out too much of the stuff that we want. And so the mixture of 50-50 works for sparkling wines, but only as an adjuvant. So we don't ever fine base wines, but if somebody does want to add it because they're concerned that it's a bit what we call, I suppose, a gelatinous year, where you become, those grapes pop out more like a lump of jelly, then a little tiny bit of bentonite into the juice or into the ferment isn't going to do so much damage. But it's always like anything with sparkling, if you're going to do something, do it to the juice. Don't start fiddling with the base wines. We want nice, lemony, citricy base wines. We don't want to start fiddling around with it at that stage so much. Okay, so let's go down a little bit more. So I've mentioned all of those things already. Fabulous. So what about proteins and what's happening with them when it comes to sparkling? Well, it's quite interesting in that there's a lot less protein synthesized in unripe berries, as I mentioned. And that's because we end up with a higher level of amino acids in cool years. So for us in cool climates where we are, then we're gonna, and we are, I suppose some people could say, making sparkling wine out of unripe grapes in some ways. So we do end up with more amino acids. And I suspect that's one of the reasons why ourselves and Champagne don't bentonite fine the base wines, don't need to. So that's something to think about where you are as well. Some of the varieties, the hybrid varieties, I have heard in some respects do have higher protein, grape proteins than, I remember some work come out of Cornell about it, higher levels of proteins in the hybrid grapes than there are in the vinifera. And so that might be a slightly different aspect for you that you might need to, but again, do it in the juice stage. There's all sorts of variations with the amino acids. Now, the amino acids, we really want those in sparkling. We want them. I mean, too many will slow down the fermentation, as I've noticed here, which takes much longer. But it'll get there. It doesn't matter. It'll do what it needs to do. So other things that add to amino acids, which is of course part of this. Not whatever that's done in the vineyard with regards to nitrogen, what rootstocks are used. Botrytis is another interesting one because it does affect the proteins in the grapes, which has a negative effect on the foaming as well. So we don't want any botrytis grapes in there. And whether we do mechanical harvesting or manual, well, all sparkling grapes are always manual harvesting, so the mechanical harvesting doesn't affect that. Going back to my point about the foam, which is why I wanted to include this, is that what's so important for traditional method foam to be integrated, to be balanced, is the complex of the proteins and polysaccharides for the bubbles there around the surface. What can happen again is if you have too much foam, that's as problematic as not having enough as well. So one issue is adding manoproteins to a wine as if to replace grape proteins. When you go to bottle by adding manoproteins, then it's not necessarily a good idea. It can cause foaming, but it doesn't, those proteins are not the same shape. They don't work in the same way as the grape proteins do. So you can often have too much foam that is way too unbalanced, too fizzy, really. But there's other things that affect foam, and these are polysaccharides, as I said. And what's really interesting with proteins and polysaccharides is that these are released and they reach their optimum at around 18 months after bottling sparkling wine, after bottling your base wine. So the longer you leave it, you're not going to generate any more. There's only so much proteins and polysaccharides you're going to release from those yeast. But it is important, and I suspect that that's why, as well, in some studies and some papers, that many of the rules and regulations sort of say you have to have at least 18 months of aging. But actually, there's quite a difference if you make sparkling between tasting a wine at 18 months of aging versus 2 years of aging. Quite surprising, really, but that will be down to these kind of compounds. As I mentioned, we did a lot of work in Canada, so all this work I'm talking about was in Niagara here, and we looked at bentonite timing, and I wanted to show our winemakers at the time why not to do it so much. And we used a Pinot Noir, a Mariafeld clone, which came from Germany or Switzerland or something. And first of all, we did the juice treatment. So it was just simply one no bentonite treatment, and then 1 gram per litre of what was called vitibentonite. So that was to 2 juices. And then we took these 2 juices, which you'll see at the top there, 2 tanks there, and then we made more treatments. So we had one that was controlled, there was nothing else added. So we had no bentonite in the juice and no bentonite at all in the adjuvants or anything else. We had another one, we had bentonite in the tirage only. So that was when we went to bottle, we added a little bit. This one was bentonite in the juice only. Hang on, I can't see, sorry. Bentonite and tirage. So there was different ones. So no bentonite, just to explain, no bentonite. Then we had bentonite in the tirage only, bentonite in the juice only, and then we did bentonite in the juice and the tirage. So a bit of a double whammy there. And what we found straight away when we looked at the foaming, first of all, was you can see here on the left, the control with no bentonite, and I'll come to the storage on that, actually had much better foam stability in it, and that foam stayed there. The bentonite in the tirage only, there was more variability between bottles. You can see that enormous error bar there. That's what that stands for. So variability between sparkling wine bottles, it was particularly interesting, but that was still only bentonite added to the actual tirage of bottling. Then we get down to bentonite in the juice, and you can see the effect on the foam was really quick. And this was 30 bottles per treatment, by the way. So we weren't just doing, it wasn't just a couple of bottles. And then when you add bentonite in the juice and in the tirage, then what you end up with was really low foam stability. That's how long the foam stayed around for. Comes back to the same question we're always asked is, does protein haze occur in sparkling wine? It doesn't have to, it can do, but it depends on how you treat the wine during the course of it and what you do, and whether your grape variety that you're using has a high level of it. But if you're dealing with it at an early stage, and because these are, let's call them slightly less ripe than your still wine varieties, they're going to have less proteins there in the first place. And that's also important to remember. And you need what proteins you have got. But like anything, if you've got, if you're using much riper grapes for sparkling wine production, then you might end up being more likely to have protein haze. I'm not going to say, I'm never going to say never, because somebody will come back to me having suffered from it someday. But I've never, ever bentonite find a sparkling wine in 22 years of making a wine. And so you take too much from it, whatever bentonite we use. So varieties, method of production, I've only worked in my research on traditional methods. I haven't used Charmat, and I haven't done any research on carbonated wine, so I can't speak on that behalf. But you're still forcing carbonation, and it takes a while to settle in, to become super saturated. But there is definitely a difference between the bubbles that are made in the wine bottle and the bubbles that are made through carbonation. There's some varieties suit one and some the other. The type of bentonite you use and the timing and the dosage is so important. And as I said, if you're going to do sparkling, do it to the juice, not later on down the line. There are adjuvant alternatives, and in fact in Niagara there was a bit of a rush on alginate for a while there. But with that adjuvant, there's a lot of work to do to prepare the wine to be protein stable and everything else before you can use it as an adjuvant. And so it's a bit of fiddly work if you're going to use alginate. The other thing that's really important and really interesting is if you've got a storage temperature of 12 degrees that you're storing your wine in for aging, and you don't use bentonite and it's left untouched, you will have different lees kind of format. It's not going to be it's going to be lighter and not stick to the side of the glass, compared to storing your wine without bentonite or adjuvant at 15 degrees. Quite surprising. It's only 3 degrees temperature, but the type of lees that form come from the temperature and the type of yeast that you're using in that second fermentation. So it's the combination of those. And there was quite a lot of work done on it in Champagne by Estacionamento something. Gosh, I can't remember the rest of the name. Richard might know the company, Enologiques. Yes, not the IOC, it's somebody else. And they just took hours and hours of photographs of all these amazing different types of yeast lees, all from just different temperatures and different yeast. So storage temperature and the yeast you use will also have an effect on foaming, actually, or whether you need to use an adjuvant or not as well. The work I showed you really briefly was, as I said, done at Covey in Canada, and the work I just showed you comes from Esther Onguta, who was at the time a master's student, but now has completed a PhD in New Zealand. And we always work with the wineries, with Trius Winery and Omafra and Guelph. And that's really all I've got for you today. And I'm hoping that you'll have lots of questions about that from there.
Great. Well, thank you so much, Belinda. That was an excellent presentation. Thank you both for your presentations. I've been fooling around with this for quite a while, and I learned a lot. And there's a few questions I know, and I've got a few of my own. So as a reminder to all the participants and attendees, now is the time to please type any questions you have in the chat box, and we will answer them. We're now going to be joined by winemaker Ann Zwink from Soldier Creek Winery in Iowa. We were supposed to also be joined by Ben Banks, but something came up. It's that time of year when winemakers are exceedingly busy. Ode, do we have any questions in the chat box?
We do have one. Hi, Anne. Good to see you. Thanks for joining us. I hope you were able to listen to almost all the presentations. So we have one question regarding non-sparkling wines. So the first question is, do you recommend doing heat stability prior to cold stability or after? And Richard, yeah, that's probably
Okay.
more for you.
So it depends. Some people do them simultaneously. What I would say is looking at your acid chemistry, if you expect, if your pH is up over 3.7, and then you cold stabilize, although your TA goes down, your pH also goes up. And so if you think about what we were talking about earlier, that higher pH might mean less bentonite efficiency. Okay. So I would say, and then also, there are a lot of different ways of looking at this. Can proteins act as protective colloids to prevent crystal growth? There's a lot of different things. But I would generally say I base it upon number one, your production constraints, and then number two, are you going to see a big shift in pH? If the pH is definitely going to trend upwards, then I would probably, during cold stabilization, I would probably suggest doing your heat stability, your bentonite finding prior to cold stabilization. On the other hand, if your cold stabilization, if your pH is below 3.6, your pH is going to drop as you cold stabilize. Then it would be worth you getting more bang for your buck with the bentonite to do it post cold stabilization. Just my 2 cents.
Thank you. And do you have any questions for the panelists? How do you do? Do you do heat stability test or you just add bentonite to find your wines? How do you do?
We just started doing heat stability tests this year. Belinda, your comments about hybrids having higher protein contents than vinifera, we have found that to be very true. We had we were using, noises. We were using large, just general recommended amounts of bentonite before, and started having some protein issues, and found that we had to increase our protein addition. Our bentonite addition by 4 times this year, just for heat stability purposes. So yeah, we did start doing stability tests after we started seeing hazes.
Do you do those tests in-house or do you send them out?
We do them in-house, but it's been a learning curve for me. The equipment is not expensive, but it's not, it seems like there's a lot of detail involved in doing the heat test in-house. So I think, a webinar on how to do those efficiently and effectively would be helpful for me.
That's a great suggestion. I'll be writing that down in just a moment. Ode, there was a second part to that question. And I was going to ask that too, because when Rich began his talk, he referred to white wines. And it wasn't to the end of his talk when he even mentioned the rosé. So I just want to confirm, of course, with what you said in mind. In general, we're worried about protein or heat instability in whites and rosés. Yes, Rich? Rich, you need to turn your sound on.
Okay, there we go. What I found was that in most red wines, by the end of primary fermentation, there's very little protein. It binds up with tannins and drops out. So, I know I said earlier, protein isn't the only factor involved, but it is a major factor. And so we don't really see much in the way of protein. And to be truthful, we run thousands and thousands of heat stability checks every year. Every now and then somebody sends in maybe a very light, cross-flowed red wine. But for the most part, it's a very rare occurrence to have anyone send in a red wine for a heat stability check. Cold stability, yes, but heat stability, no. And once again, based on the work we've done with the chitinase and thaumatin-like protein ELISA tests, we just don't see that much protein in red wines.
Okay, and I was just mentioning that I was asking about rosé.
Well, rosés can be. I think someplace on our website we have a white paper on the difficulties in heat stabilizing rosés.
Okay.
Rosés can be amazingly difficult, and this is why that I use the reference of the person trying to heat stabilize a rosé. Sometimes it's 10, 12, 20 pounds per 1,000 gallons of bentonite, and that will just strip the wine. I mean, we had one year, I think it was 2015, it was a very hot year, and all the rosés were remarkably unstable. Okay, we know from just years of doing heat stabilities for clients that there's definitely a vintage impact on heat stability. I mean, there are years where it's really bad, probably for whatever reason. There's more protein, maybe more polyphenols, but certainly in rosés you have skin contact, so you have more polyphenols than you would in white wines. Okay, one of the things I've found is that in some cases, using a broad spectrum polysaccharide degrading enzyme, something like I hate to mention anyone's names, but anyways, they just say a crude pectinase that has some glucanase in it and things like that. You'll get, and you treat the rosé with that, and then you'll have a much more linear bentonite finding trial. I mean, sometimes with rosés, we had bentonite, at 5 pounds, or excuse me, at 0 pounds, you're at 60 NTU, and at 1 pound, it drops down to 38, and then at 2 pounds, it bumps back. It's just, they make no sense as far as the results. But we found that removing some of the pectin and polysaccharide component can help stabilize that. And give you a more linear response to the bentonite. So I believe that once again, we certainly at this point don't have a complete understanding of that colloidal formation, but I think with rosés having skin tannins in there, as well as perhaps more pectins or whatever, I'm not sure that would, but yeah, if you've got rosés and you're having problems, you're not alone.
Okay, good.
And did you have a question?
I think it was similar. Richie made a comment about how vinifera specifically the tannin binds up the proteins, and so you don't really see an issue. And so I'm just curious, if you've had a lot of hybrid wines coming in that don't have a lot of tannin, are we having protein instability issues?
Yeah, that's a really good
more
question.
Commonly here.
I think low tannins, certainly you could have more problems. I also, we've only done a little bit. I didn't bring it up because we've only done some work with a few non-vinifera varietals, but they have elevated levels of protein. The few that we've looked at, and that would make sense because most of the non-vinifera varietals are generally more pathogen resistant. And disease resistant, and so therefore their constitutive level of those pathogenesis proteins may be expressed at a higher level. Once again, I can only say I think we've looked at maybe 2 or 3 varietals, and so I have seen that, but not enough that I could make a slide out of it. But the fact that if you've got higher levels and lower tannins to precipitate them, then that would be maybe something, if you've got a red varietal that's low tannin and you just want to send it to me, I won't charge it just to look because I'd be curious to see. I'd be happy to run the chitinase and thaumatin-like protein just to see. It'll be another data point I have. So just send me one, okay?
And if I can jump on that topic, because we have been trying to evaluate how that pathogenesis-related proteins are impacting the low concentration of tannin in wines, but it doesn't seem to be very well correlated. So I think that's two issues. So we have a lot of proteins, but at the same time, we don't have a lot of tannin. And sometimes it will be related, but sometimes it won't. And it's because we did an experiment where we tried to use bentonite to remove proteins, trying to get more tannin out of those wines, but it wasn't improving the concentration of tannin.
No,
And
we're
so,
actually we're doing a project right now with Steve Price and his vineyard up in Oregon, where Steve, if you don't know Steve Price, he's a really great phenolics viticulture guy, and he's got a beautiful Pinot vineyard, but he's always doing little experiments. So I had him go out and abuse some clusters, so we would really ramp up, physically abuse them, and then we would ramp up the pathogenesis protein production, and then we're going to look at tannin extraction, because you're right. There's been work published showing that if you have high levels of pathogenesis proteins in the skin, they actually bind up the tannins, so you don't get as good an extraction of the tannins. So that's something we're looking at right now. So we have the tools. We just have to. We're not a research group. We're a production wine laboratory, and so we try to squeeze in little research projects when we can.
I'm gonna try to help you squeeze more research out of your capabilities whenever we can.
Yep.
And I just want to confirm, both for Rich and for Belinda, that yes, indeed, these new American hybrids, if you will, especially the University of Minnesota hybrids that I deal with on a daily basis, as we mentioned, they certainly do have higher proteins, and some of them in certain years, especially very warm years, which we've had, also have very high beta glucanase content. And that has always also presented challenges also in final filtration of these wines. And so I've had to suggest filtration enzymes in relatively high amounts for people who are having a problem with these increased beta glucanases, especially in late harvest style wines. All good stuff. Do we have any other questions?
we have another question, but that's more for cold stabilization. So that's not exactly for protein, but that's more for tap rate. So what's your opinion of chemical methods of cold stabilization? So that's
I think that, people have been, I think Lafort was the first to come into the US with CellStab, which is a carboxymethylcellulose. And that was widely accepted and does a really good job. The biggest problem with using these, what I call additive technologies, is that you're not doing traditional cold stabilization, so you're leaving all of the tartaric acid in the wine. And if you have high calcium, then you're going to end up with calcium tartrate precipitates. So in California, down in the Paso Robles region in the Central Coast, they have high calcium soils, but it's also very hot there, and energy is expensive in California. So everybody decided to use CellStab when it first came out. But nobody read the very first line on the CellStab worksheet, which said, check your calcium. No one read it. And because it said in the juice, check your calcium. And people went down to, we're making wine, I don't need to read about the juice. And so they didn't check it. And so where we used to get maybe one calcium tartrate precipitate a year come to the lab, we had hundreds. And so all I would say is these products work really well. You have to make sure the wine is heat stable. If it's not heat stable and you add any of these products, all of a sudden you're going to have a big tank of cloudy wine. If you have calcium, you can end up with problems. But I will tell you that every vendor has, and it's very confusing to use them. That's the other thing, is that every vendor has different criteria for what we call pre-addition screening and then post-addition verification that it worked. Different methods of conductivity, different alcohol or calcium, or most everyone's the same on heat stability, going to be below 2. But these work very well, and people like them because it saves a lot of money. So that's just, I mean, in Minnesota, you can just open the door and wheel the tank out and cold stabilize it. It doesn't cost you anything, right?
I try to teach people this all the time, that we have free cold stabilization. And
Exactly.
again, not to belabor the point, most of our wines do tend to be rather high in grams per liter of acidity and relatively low in pH, especially the whites. And so the traditional cold contact, cold stability helps with that, helps drop out the excess tartaric acid.
Yeah.
I will mention again, as you mentioned, that sort of magic break point of pH about 3.65, where above that, with cold stabilization, your pH can rise, and below that, it can drop. I always remind newer beginning winemakers here that they're invest in a good pH meter, take care of that pH meter, know how to use and calibrate that pH meter, and for God's sakes, write down your results so you can compare wine to wine, season to season, year to year. So you cannot get away from pH. If I had one tool to make wine by, it would besides my tongue, it would certainly be a high quality pH meter.
And I have a question for you, Belinda, about the calcium. So talking about the amount of calcium that might be challenging, you mentioned that the water of UK is pretty rich in calcium. Can you elaborate a little bit more about that challenge of having high calcium content in water?
Yeah, it's actually something I forgot about, and then 10 years in Canada came back. So if we're washing our new tanks ready to put the red in tomorrow morning when we press, and there's so much calcium in our water that it leaves a sediment. As the water's dried and disappeared, it leaves a calcium sediment on that looks a little bit like limescale, but it's white, and you can just rub it. And I thought, oh, maybe they've sulfured everything, and with powder. But going round, no, it's a big circle on the bottom of the tank, and these are new tanks that have never been used, and you wipe it across, and it's calcium. It's something, we have calcium crystallization issues here, calcium tartrate crystals here. But then we have very low pHs here. You'd hate the chemistry that would be over here. You'd hate our tartaric acids, horrendous. And then we've got this other thing where this calcium. So as soon as we're using using the what we call hard water, which means that in some places here in the UK it can be over 300 milligrams per litre. That's huge amount of calcium. And so if you're using, and in some places in the countryside is between 100 milligrams per litre and around 2. So if you are using water for all your additives, whether that be, if you're not using SO2 in liquid form, for instance, if you're adding nutrients to it all the time in that 10,000 litre tanks, we're adding calcium all the way. And it's something that our own industry hasn't realized. We go, we've all suddenly got calcium problems, and it's grown more and more as the industry's grown. And going, well, how do we, we're just because the juice is one calcium level, that's just the start of the additions that we're making. So that's new to me as well. And that's something this is the first vintage back in England for 10 years for me. So it's like, oh no, we can't even use the hose from the winery to clean the tanks, because that gives us a whole, sort of this deposit on the bottom of the tanks. So it's an interesting one. I didn't expect that. I thought of lots of other problems that we'd be dealing with when I came home, but no, it's quite an interesting one.
Thank you. And I have another question that's related to your presentation where you mentioned bentonite versus ketosane versus PVPP. So have you tried ketosane plus bentonite for fining?
I haven't. So the thing with chitosan is, when we published a paper actually earlier this year about using chitosan on juice, we didn't use it on, well, we did use it on wine, but we were looking at a specific family of compounds, which were the compounds for Maillard reaction. So we didn't include protein measurements in that. So we use the chitosan in the juice, and it's really interesting because you can cut down on the amount of SO2 that you use in the juice by using chitosan. And the other thing that the chitosan did was pull out phenolics out of the juice. We settled it overnight with chitosan in it, and then just racked off as we would normally do, rack off the solids the next day and fermented. And the juice and the wine had far less phenolics in it. And so that's been published. But there's so much work to do on chitosan because it can do so much more than we think it does. And because there's so many different types and so many molecular weights of it, it's really a fascinating compound. By leaving it in wine for just 4 months, base wines, we activated and made all these Maillard reaction compounds start happening, which don't usually happen until much later when you add sugar into the wine after, in the dosage. And so we sort of, by adding it and leaving it for 4 months, we had accelerated the flavor development in the base wine. So the whole world of chitosan is just really only getting started, and there's so much work that can be done in that area. As I said, with there's so many different types now, and different timings, different amount of additions when you're doing it. What's the reason what the purpose is you're targeting, and so yeah, anybody who wants to do chitosan trials, I'm in.
Fantastic. It's constantly amazing to me how every year or 2 or 3 there is some major new nugget of practical winemaking, something that I've been counting on for 25 years, all of a sudden changes, or there's a better solution, or even a vegan solution, instead of using animal proteins. One of these things that I just learned about today from Rich's talk, and if you would give us a few more minutes, Rich, about it. I've been out of commercial winemaking for about 10 years now. I was in it for 20. You talk about using bentonite in the juice before fermentation, being able to heat stabilize wine. Can you talk more about that? I've always done it after fermentation. I've never really heard of doing it before fermentation, in rare cases only.
Yeah, there's actually, I think most of the big vendors have bentonite specifically designed now for using in juice. And I think once again, they might not take it all the way to being heat stable, but they remove a lot of reactive protein. So then when the time comes, post fermentation, when they're getting ready to bottle and they're going to heat stabilize, they just need a little bit of a dusting of bentonite and not maybe a heavy hit. So it seems to be very popular. And once again, the idea being that bentonite is, got a net negative charge, binds anything with a positive charge. So when you put something that's, very not specific as far as its binding capacity into wine, then you're pulling out a lot of things. And so for that reason, I think many of these components are either, like I said, glycosylated aroma compounds, or maybe color compounds that are still bound up in skin or whatever. They're not going to get pulled out if you're making a rosé or something you pressed off. So it seems like a good way to do it. And so there is a growing use in the industry of bentonite in the juice.
I would one of the last jobs I had in commercial winemaking was for a small winery owned by a very, very large company, and we dealt with a lot of machine picked whites from the Central Coast, and we used to treat the juice with 1 pound of bentonite per 1,000 gallons, and a bit of PVPP, and I was amazed to find how quickly that could settle, even as short a period as overnight.
Mm-hmm.
That juice was racking very clean the next morning. Do you agree that even just a 24-hour settling period after you've added a small amount of bentonite to the juice is sufficient, or do you think you need to let it settle for 2 or 3 days?
Well, I mean, these people are adding to juice, even adding it in as the fermentation is starting. So they're not necessarily, so there's still some residual in there. So I don't think it really impacts fermentation at all. If you look under a microscope, the stuff I call bentonite dust, which is just super fine particles. Anything that's floating around is giving yeast and bacteria something to anchor onto, and they like that in the case of getting through ML or even primary. That things seem to work better if the more stuff you have up to a point. But yeah, I don't. The people that I know who are using it aren't being super cautious about racking off of all of it.
Sure.
But you're right, it does drop pretty quickly in juice if the juice is pretty clean.
And I've just got one other comment I want to make to all of you and to all the attendees before I forget, because again, I get questions from new winemakers, beginning winemakers, who have heard that bentonite can strip color, flavor, character from wines. I've worked at a winery where we bottled wine that was supposedly heat stable, and within a month, a few thousand bottles of wine had a haze, and we sat there pulling corks on a few thousand bottles of wine and pouring it back into a tank. The whole time, the winemaker muttering under his breath, "If only I'd used another $10 worth of bentonite." I've never seen or heard a winemaker say to themselves, "Gee, I wish I'd used less bentonite." I've heard them say, "I wish I'd used more," but I've never heard someone say, "I wish I used less."
Yeah.
Would you tend to agree with that?
Well, I think that, 2 to 3 pounds doesn't seem to have too much of an impact. But there are wines, we have years that are really bad. It's just kind of weird feedback. We have years that are really bad, where people's, control heat stability is coming in at 200 NTU, when we do the trial. And so there are just years where, in order to achieve that number, which I said you don't always have to chase, but if you have to ship wine to wine club members in August who live in Texas, you need a certain level of stability. And so in that case, you see numbers coming back where you need 8, 10, 12 pounds, 1,000 gallons of bentonite. And at that level, you're definitely stripping a rosé, you're stripping color, you're stripping all kinds of aromatics. So there's no doubt that occurs. And that's why I think we're seeing more people interested in putting some up front in the juice. So maybe they have to add less later.
Makes good sense. Ode, I see a statement here, and then I'll read the other question if that's all right.
All right. So yeah, we have a comment about the product from NRTs, which is the Clairol SP, that is recommended to be added to the juice. It contains bentonite, PVPP, and caseinate.
Well, I mean, every vendor probably has a product. I bet Lallemand has one, Guzmer probably has one, Lafort probably has one, where they mix all these things. So as Drew was saying, they were adding PVPP and bentonite. Okay, so the PVPP is gonna, if there's any browning things coming out like that, you're gonna get rid of some of the browning compounds. The bentonite, you're gonna remove some protein. Casein does a really good job of binding up off aromas and things like that. I find some of the products that contain casein, there's one that contains casein and bentonite. It actually can remove dimethyl sulfide, which theoretically you can't do that, but you can, and diethyl sulfide. So I would say that something like that product, where you're kind of throwing the kitchen sink at it, could probably fix a lot of problems. If you had a juice that maybe had some, botrytis and lactase activity, you're getting some oxidation, maybe some off characters, yeah, that would probably work. But I also tell people that, remember, the person you're talking to is trying to sell a product. Okay. And I say that because we don't sell any products, right? We just do analysis. But the point is, there's a lot of claims out there. But what I would say is, you try it, and if the problem is no one does research. So if you were to take a juice from a particular block that had issues and treat half of the juice with the product and not treat the other half of the juice, and then actually follow those through, then you'd have the answer. But that doesn't happen a lot in the real world. So I would just say, in regards to products, there are certainly a lot of products out there, and they can work and do work in certain circumstances. And I'll just leave it at that.
There's a question here about what effect, this is a good question. What effect does bentonite have on the use of enzymes? Again, I'm thinking this is about white juices. Again, I worked for this winery in California. We would add enzyme immediately, right at crushing, during crushing. And then once everything was in, it was a closed tile press. Then after the enzymes, we would add sulfur, and then we would wait and then make that bentonite. We gave some time for the enzyme to work before we added the sulfur. Anyway, the question is there any effect that bentonite has on the enzymes? Are there any issues with bentonite and the finished fragrance of a treated wine?
I mean, you know, if the enzymes you're adding have an isoelectric focus point where they don't have a strong positive charge in the wine, then there shouldn't be an interaction with the bentonite. But if they do, yeah, I mean, and then if you get something completely indiscriminate, like when people have issues with a really high botrytis load, they're going to, you know, add enzymes to pectinases to break everything open and release all the intracellular lacase, and then they're going to hit it with a sacrificial tannin. But if you put an enzyme in and throw the tannin in, you just immediately precipitate all the enzymes. So,
Right.
so that doesn't work. But with bentonite, I think it really depends. And you know, if you go buy, you know, a commercial pectinase, I don't know if anywhere on the label it tells you what the isoelectric focus point is on that enzyme. But that would, I guess, that would be the thing. We always think that protein and bentonite interact, and there's always that possibility, but it really depends on the net charge on the protein at the wine pH or the juice pH. And as far as stripping aromas, yeah, that's, I think you can line up a whole bunch of winemakers and they'll all agree that if they've added, you know, more bentonite than they wanted to make a wine stable, that the wine has suffered as far as aromatically.
Is it what you would say, Anne?
I think maybe I could see that happening with more delicate style grapes. But we haven't really had an issue with that with our intensely aromatic hybrids. But the only ones that I've really had to add a huge amount of bentonite to are like in the Frontenac family, sometimes La Crescent family, and those tend to be very, very aromatic. So it hasn't really caused too much of a loss for us.
There's one more question here from Jay Stoger. And one of you can clear this up. He says, if I have a pH between, I'm assuming this is a finished wine. If I have a pH between 2.9 and 3.4, does this make bentonite useless? I don't think it makes bentonite useless, but again, you want to talk about the relation of pH and bentonite?
Well, I just think that it's about the proteins in your wine and what the isoelectric focus point is. And most of the PR proteins have are going, as your pH gets lower, 2.9 to 3.4, you're going to have a stronger positive charge on, because these things have PIs in the 4 to 6 range. So the lower your pH is, or I should say, the greater the distance, the more negative your pH is to your isoelectric focus point, the stronger the positive charge you're going to have on your protein. So it's going to be more reactive with the bentonite. So at lower pHs, I would expect that you're going to have a better bentonite efficacy because the proteins have a stronger net positive charge.
Well, there you go. So the answer to that question is no. Bentonite is not useless.
Whereas if you have something with a pH of 4.5, 4.6, it happens, then that might, you might not get a lot of efficacy from your bentonite.
Sure. Good stuff. And Belinda, I just wanted to say, I thought it was an interesting solution. Who came up with the idea of using 50% calcium and sodium bentonite to address your high calcium level in the UK?
That came from that's actually been around for a long time. That came from Champagne, and there's products that are made in Europe that are 50-50, so you can buy them like that as opposed to knocking them up yourself. Yeah, which you could do, but
well, that's good to know. We do have a broad reach, both with this series and with these hybrid grapes. They're starting to grow these, of course, in eastern and central Canada. We're hoping to get them soon into Scandinavia, northern Europe.
the peewees.
Say again?
The peewees, they're called over here. the German name for them, the disease-resistant varieties you're talking about. Yeah.
Yeah, yeah.
Yeah, the camp is split on at the moment, all over Europe. Actually, it's quite interesting. Quite interesting. It's pros and cons for everything, though, isn't there, in life? And the disease-resistant varieties are no different. It causes, yeah, we call it like Marmite. Some people love them and some people hate them.
I hate them. I keep reminding. I'm hoping both of you at one point have heard of the University of Minnesota red grape Marquette,
Yes.
and it's probably the most widely planted. I was in the Vitinord conference in Vermont last December, and there were some wonderful examples of Vermont and New York and New Hampshire Marquette and blends of Marquette and Petit Pearl. They have been widely embraced, but there was some from the people who were, as I say, from Northern Europe. They were sort of evenly split about whether they liked them or not.
So,
It's nice to have built-in disease resistance. It's nice to have extreme cold hardiness, of course.
yeah.
I was recently contacted by a winemaker in the Okanagan Valley in British Columbia, and they had a horrible, very low temperature event.
Yeah.
I believe it was in April or something, which never happens, and it killed their Pinot Noir. And all of a sudden, this winemaker became very interested in these cold hardy grapes, and maybe do I know anybody in that part of the world who's growing them, and where can he taste some of the wine? So
Yeah. There's some in,
we get interest from all sorts of places.
yeah. There's some in Ontario as well. There's
Yeah.
Marquette is allowed on the BQA list in Ontario.
Yep, that happened just a few years ago.
Yeah.
Well, I see we've only got a few minutes left. Oh, do you have anything else to tell the audience?
We still have one last question, and I'm going to share my screen at the same time that I'm asking that last question. That's related to a product. Let me see if yeah. All right. So do you have any thoughts on Kieselsol for dropping protein as an alternative to bentonite? Have you heard about that product?
Kieselsol is a silica, so it's widely used as a clarifying agent, but not specifically for protein. And I think it cost a lot more than bentonite. But I don't know of anyone using Kieselsol specifically for heat stabilization, protein removal. And once again, I know there's a charge base, but I think Kieselsol is a silica gel or silica product.
Yeah, it's a clarifier,
right?
Not really a protein
Right,
stabilizer.
right.
Yeah,
It helps things settle out and clarifies the wine, but I don't know of anyone using it. I mean, I would say that if you had a wine that was really turbid, and if you use Kieselsol to settle and clarify the wine, then the wine would probably respond a lot better to the bentonite fining treatment, because all of that stuff that's floating around in the wine is going to interact potentially with the bentonite.
I think the majority of Kieselsol is for the home winemaking segment. I don't know any commercial wineries using it. And you don't use Kieselsol, do you? Okay.
No, we occasionally have clients that use it for something very specific, but once again, not necessarily for protein fining.
All right. Well, thank you very much. Aude, it's up to you.
Yeah, thank you very much, everyone. Thank you, Belinda, for staying that late with us.
Yes.
Thank you, Richard, as well, for your great presentation. Thanks, everyone, for attending that webinar. And so before closing the webinar, I just want to make sure you are all aware of the next webinar that's going to be on November 14 about waste management. So we're going to talk about water waste management, other byproducts that can be developed in your wineries, and also please make sure you have you take the survey. So you can either scan the QR code that you see here or use the link that I shared on the chat box. That's really just to help us improving our webinar series. So once again, thank you very much. And we look forward to seeing you all in November 14.
And Belinda, if I ever make it to the UK again, I will certainly take you up on your invitation. And likewise, if you ever find yourself in the Midwest, come see Ode in Iowa or me up in Minnesota. I can show you a field of 12,000 new individual vines that we're evaluating.
Fantastic. Okay.